Hello everyone. I made this video to provide you with feedback on the first exam that we took for this semester. So let's get right to it. The first question asked you to draw an acid-base distribution plot for arginine. So a good way to get started for that is to draw all the structures of arginine from low pH where all the functional groups are protonated. The amino group, the carboxyl group, and the guanidino group are all protonated, and then you take off their protons one by one depending on which one is least likely held. So the carboxylate proton comes off first, the alpha amino group proton comes off next, and then the guanidino group comes off last. So here we have 3 protons, the overall charge is +2. We pull off a proton here, and we have a +1. There's our zwitterine HA right here. And when arginine is completely deprotonated, that's A-. So we have 4 species, so we need 4 curves, and this is what the plot looks like. So there should be 3 crossing points at the concentration that's half the formal concentration, because that's where the pH equals the pKr value, or pKa1 or pKa2 value, depending on which one it is. The zwitterion is over here. This arrow right here indicates the pI, because the pI is that pH at which the zwitterion concentration is the highest. So that would be the peak of this curve right here. And that would be in between the pK2 value for the amino group and the pKr for the guanidino group. So the next question, questions 2 and 3 went together. So here we're looking at our patient's hemoglobin, and I set this up purposely so that it would not be too difficult, I hoped, for you to see the differences. I highlighted in green here the differences, and we can see that there's this long extension that our patient Li Jing has in her alpha globin chain. One of the things that the investigators did when they purified the hemoglobin and wanted to analyze it, they didn't know what they were looking at until they did purify it. So they did a trypsin digest. We can see the trypsin cleaves after arginlyse. So that was given in class, and you were told to memorize these. So if I go back to my normal versus Li Jing, I really just need to look at the differences. So R is arginine and K is lysine. Those 1-letter codes were given in the appendix. So if you just see what's different, there's an arginine right here. So trypsin will cleave here. The next arginine is here. There's an arginine here. This arginine actually will not be cleaved because if the following residue is proline, these enzymes don't work because of the configuration of proline. So that's not going to cleave. So what we can see is that we get 3 more fragments. Some people actually went through the whole thing and figured out the number of fragments. You didn't have to do that. You just had to know that you would get 3 additional fragments from the trypsin digestion compared to the wild type. So that helped the investigators see what's going on, is that they if you just had a point mutation, you would get a different fragment, but they got 3 extra fragments, so they knew they were dealing with a longer protein. So if you look at the DNA, what would produce that? So what would happen is, again, you have to go back and look at this. The next amino acid right here, it's supposed to end at an arginine, but there's a Q here, and Q is the 1-letter code for glin. So you'd sort of do this backwards, in that you would put in a glin codon right here. And if the normal stopped here at the arginine, the next codon must have been a stop codon. So what happened is there was a mutation in her DNA that changed a stop codon to a glin codon. And what happens then, if the stop codon is not there, then the ribosome just continues to translate all the way through what normally would be the 3' untranslated region of the messenger RNA. You would eventually get to a second stop codon. Lots of these messenger RNA genes have more than one stop sign, just to make sure that the ribosome stops when it's supposed to. That's why we got a longer protein, is because we sort of blew through that stop sign and didn't stop until we got to the next stop sign. Note that this is the DNA on the coding strand, and it was the DNA that was requested, not the messenger RNA, so you would report T and not U. The next question could be a little bit tricky. It's just kind of a puzzle to solve, and I'm going to give you a long, complicated answer, but you didn't really actually need to know that much in order to get this right. So we have hemoglobin A, that's normal hemoglobin, and hemoglobin A2, which is a minor form of hemoglobin. About 2% of all adult hemoglobin is this A2. So you've got mostly A and A2. So a normal person would have these two bands right here. But she has two extra bands. She has what we call hemoglobin 1 and hemoglobin 2. So what are these? Well, if we look at this isoelectric focusing gel, the negative pole is right here. We would have loaded the hemoglobins on at the origin, and then the hemoglobins would have traveled to the positive pole. And they would have stopped when they got to where pH equals Pi. So this is pH 7 right here that I have indicated here. Then hemoglobin 2 has a Pi that's less than 7, and hemoglobin 1 is actually more basic, has a higher Pi than either A or A2. So it's very basic. So we just have to figure out what kind of combination would give us something more basic than normal, what kind of combination would give us something more acidic than normal. So again, if I look here at her hemoglobin, again, look at this extension. This is what makes her alpha chains different. We already went through this and saw that we had arginines, and we didn't have any lysines, but we had those arginines. But if we look and see, do we have any aspartates and glutamates? If you look here, I only see one glue, and I don't see any asps, but I see the arginines I pointed out before. There's an arginine there, there's an arginine there, there's an arginine there. So this extension has extra arginines and not extra glutamates or aspartates. So this extra chain is basic. So her alpha globin chain is going to be more basic than normal. And remember, her beta chain is normal. So if it's going to be more basic, then if a normal hemoglobin A beta chain is a Pi of 6.74, now you would have to go to look at question 7 to see that, but even you didn't actually really have to do that. You were given that the normal alpha chain Pi is 8.73. So this extension means that the Pi has to be higher than that. Now, I got this number because I actually put the sequence through an online Pi calculator, which you obviously couldn't do. But that doesn't matter. You just needed to know that whatever it is, it was more basic than 8.73. That's all you had to know. So I did actually go ahead to question 7 and I looked at the normal beta chain. So if this is normal alpha and normal beta, then my normal A has to be some kind of average between the two, and that's where it is. So I'll just go to hemoglobin 1 first. But that means that hemoglobin 1 has to be that alpha 2, beta 2. Her beta is normal, and that extension is more basic. So hemoglobin 1 has to be alpha 2, beta 2, but with abnormal alphas. So that means that whatever is here has to be more acidic. So the only thing possible here is really beta 4. And again, you can go to question 7 and you can verify that. But again, I'm giving you sort of a more complicated example. If this is hemoglobin A, then hemoglobin 2 is more acidic. And we do that by getting extra beta chains. Because remember, she has extra beta chains. She has this deficit of alpha chains. And hemoglobin 1, again, I just averaged the two. But again, you didn't need to know that. You just needed to know that it was more basic than hemoglobin A. So I presented it to you in a way that's maybe more complicated than you need to. You just need to figure out which one's more acidic than normal and which one's more basic than normal. That's really all you had to do. So what that means then, to answer the question, is that the hemoglobin 1 has 2 abnormal alpha chains and 2 beta chains that are normal. And the way I figured this out was that it's more basic than normal, and the hemoglobin 2 is more acidic. So, so that's 4 beta chains. So some people put alpha 4 and hemoglobin 2 is alpha 2, beta 2. PI-wise, that works, but it doesn't really work because we don't have extra alphas, but we do have extra betas. So beta 4 is going to be the more likely possibility. So the next thing, so that's all the information I gave you, except for this table. So you want to write a structure function argument for her symptoms. So if we look at her symptoms, we can see that her hemoglobin is low. The amount of hemoglobin per cell is low. Now, she has a normal amount of cells, so her cell count's fine. But in that cell, there's a lot less hemoglobin. And if you add up sort of the hemoglobin grams per deciliter, it's going to be low. And that's because the number of cells is normal, but the amount of hemoglobin in each cell is low. And again, she's got extra betas, and she has this deficit of alphas. So we don't have a whole lot to go on. So that means that we have to go back to biochemistry, sort of first principles. We have a profound change in the primary amino acid sequence of her alpha chain. And it's not just an extra amino acid or a change in amino acid. It's this huge, long extension at the C terminal. And that is going to cause the protein to fold way, way differently than it would have if that extension were not there. So that means, we learned this in class, that primary amino acid sequence affects the folding, which affects the secondary structures of the positions of the alpha helices and overall tertiary structure. And we can see from the isoelectric focusing gel that tetramer assembly was affected too, that beta 4s are getting together, and that doesn't normally happen. So you wanted to make a strong structure function argument using the principles that we learned in class, and that's really all we have to go on. And the question asked for that. Beyond that, we don't exactly know what happened, but we do know the structure impacts function. So it could possibly affect the structure in the vicinity of the oxygen binding site. And it will probably interfere with protein function, oxygen binding to hemoglobin in the lungs, delivery to oxygen to the tissues. We don't have any of that data. So we just have to hypothesize that would happen. Really, all we know is that we have less hemoglobin. So you could cite the values that I just showed you in that table. So these symptoms, we have less alpha chain synthesis, or maybe the synthesis is normal and they just get degraded. But the point is we have less alpha chains. We have an abnormal tetramer assembly. Probably the beta 4 doesn't have the same cooperativity as alpha 2 beta 2. Again, we don't know that. We just know that she has low hemoglobin. So we know she has less hemoglobin, so that means that oxygen delivery to the tissues has to be compromised. If she doesn't have that much hemoglobin, she's going to bind less in the lungs. She's going to deliver less to the tissues. But we don't have any extra data to support that. We don't know anything about the stability of the R form versus the T form in this patient. We don't have that data. We don't have data about allosteric effectors. We don't have oxygen binding data. We don't have any of that. So we really can't say that in our answer because we don't have that data. All we know is that the hemoglobin concentration is low, and we can go back to our structure function arguments to say the structure isn't normal, and the secondary, tertiary, quaternary structure are also not normal, and that has to impact function. So it could be that the abnormal hemoglobin distorts the red blood cell shape, but probably not that much because the cell count is normal. So abnormal cells are removed by the spleen. So if there's not an increase in that removal process, then the shape of the cells is probably normal or close to normal. So some students mentioned oxidative stress, but again, that can't be too great because the red blood cell count is normal. So that's probably there, but it's not too bad because the red blood cell count is still normal. So in your test taking strategies for the future, the question does ask for structure function, so that needed to be there. Also, it's a 10-point question, so if you just write 1 or 2 sentences, you're probably not going to be able to cover the kinds of concepts I just discussed just now. And you want to make sure that you interpret the data. I got a lot of people just sort of writing down what was in the background. So you need to bring something else to the question besides what's presented in the background, even though the background, of course, will inform your answer. And as this is a biochemistry class, we need to have some biochemistry concepts that we learned in class, and I need you to demonstrate to me that you've learned those. Okay, let's go on to question 7. So question 7, our patient, Rashmi, has inherited an abnormal hemoglobin from each parent. So here we're focusing on Rashmi's mother. So the physicians collected red blood cells and lysed the cells and then separated hemoglobins. And you were given that the alpha chains are normal, but the beta chain of one hemoglobin has a PI of 7.86 in the beta chain, has a PI of 6.73. So here we can easily tell which is which because there's a glue to lyse substitution. So what that means then is that if the hemoglobin A is normal and has a glue residue, that's acidic. And if there's a lyse substitution, that's basic. So that's a big enough change that you're going to get a big difference in PI. So the acidic change is going to give you a more acidic PI, and the basic residue is going to give you a more basic PI. So this comes down to knowing the structures of the amino acids, but say you were in the exam and you get a little nervous and you couldn't remember. Well, we have lots of resources. If you look at the back of the appendix and you look at glutamate and you look at lysine, and you can see the pKr values, and you can see that the pKr of this glutamate, so the glutamate residue in the protein, is much orders of magnitude lower than the lysine. So that's another resource to help you out. Okay, so we have these two hemoglobins because the patient, this isn't the patient's mom, so she's going to have one abnormal hemoglobin and with abnormal beta chains and one normal. Given this, I chose a pH that was midway between these two. You didn't have to, as long as the pH was greater than 6.74 and less than 7.86, I counted it as correct. But it couldn't be less than 6.74. It couldn't be greater than 7.86. I chose phosphate because that's easy. What happens here is that if I choose 7.3, and this will work with 7.3 because the pK of this is pKa2 is 6.8, so it's within 1 unit, which we learned in our POGL activity. And if I choose this pH, the hemoglobin A is going to be negatively charged and the hemoglobin E is going to be positively charged. So that way, because of the abnormal beta chain, so that way I know I can separate them. So if you look at the chart that I gave you in the appendix, you have all of these buffers from our book to choose from, but the only ones that would work were these: phosphate, ACEs, imidazolium, paranitrophenol, and HEAPs, because those are the only ones that have a pH greater than 6.4 and 6.74 and less than 7.86. What you're given here in this table is you're given the weak acid structures, and the ionizable proton is shown in red. So, but I asked for the structure. So H2PO4 minus is the weak acid, HPO4 2 minus is the conjugate base. If you drew this structure, you would just draw an H instead of an H2, and then that wouldn't be positive. Here again, leave off the H, and then that's neutral. Here, pull off this proton, and this becomes O minus. Here, pull off this proton, and this becomes a neutral nitrogen. So any of these that you wanted to use was acceptable. Remember, a buffer has two components, a weak acid and a conjugate base. We spent a lot of time in our POGL activities talking about this. Some people wrote one species, some people wrote 3 species. I was a little curious about that. I had hoped I had convinced you that buffers need two components, a weak acid to mop up any strong base and a conjugate base to mop up any strong acid that you might add to your buffer, in this case, the blood. Okay, so now we can separate this. So remember, Rashmi's mother has normal hemoglobin A, and she has a hemoglobin E, which has a normal alpha chain and a mutated beta chain. Before I showed you that if hemoglobin A, if it's adjusted to pH 7.3, its PI is less than the pH, so it's going to be negatively charged. Cation exchange chromatography involves these carboxy methyl groups. They are negatively charged. This is cation exchange. Cation exchange is named, remember, for what binds to the bead, not the bead itself. So if hemoglobin A is negative, it's going to be repelled. It'll come off first. And hemoglobin E will be attracted, so you have to wash a lot of buffer through it, and it will come out last. We monitor this by elution volume as the proteins come off the column. You could say fraction number, you could say time, that would be okay. On the y-axis, we typically monitor the concentration of the protein, and we do that because concentration is proportional to absorbance at 280 nanometers. This is the lambda max according to Beer's law. Concentration is proportional to absorbance, so we would get our nice peaks here. It is important to note that we were separating whole hemoglobins, not just the beta chains. It's the beta chain that makes these two hemoglobins different, but we were separating the whole hemoglobins. Okay, so for question 8, we wanted to calculate some Y values. So you have to go and look at your background and look at your resources. And here we will need the P50. So Rashmi's hemoglobin has a higher P50 at 30.2 millimeters of mercury, and the volunteer who's healthy is 25.6. So if I use these values then, and the N values I'm provided, I can obtain these Y values. And just to give you my calculations, again, here's the normal person. Their N value is 3. That's their P50 value, so you'd solve it for 20 torr and 100 torr. And this is our patient. She has a higher P50 but a lower N, and these are the values that you calculate for her. I use torr. Torr is, that's just what I'm used to, but torr and millimeters of mercury are the same. Millimeters of mercury, if you remember from general chemistry, that long tube that was designed by Torricelli. So he, of course, measured the height of the tube in millimeters of mercury, not knowing at the time how toxic it was. So after Torricelli, we just named millimeters of mercury torr after Torricelli, so they're the same. It's important to have your units. A lot of people left this out. I thought it was pretty clear that you all have to have your units. Y doesn't have units, and we can see that these units will cancel in the numerator and the denominator. So please show that in your calculations. Okay, question 9. I don't have the crystal structure. I don't even know if it exists of this hemoglobin E, where the glutamate is mutated to a lysine, but I was able to just pull up on Pymol from the protein data bank, just normal deoxyhemoglobin. And here's the glutamate that I'm circling right here in orange. And nearby, you can see that this is an arginine, which is shown in yellow, and you can see a histidine, which is shown in slate blue. So this is the T form. This might be one of the ion pairs that stabilize the T form. I actually don't know that. I'd have to go back to Protein Data Bank and look at the R form to see if those interactions are broken, and I didn't do that. So it's possible that these are one of the ion pairs that stabilize the T form, because this is the T form. So what I wanted you to do is I presented you with the 3-dimensional image, and I wanted you to draw a 2-dimensional image. So if I look at my beta chain, here's my glutamate, there's my glutamate side chain, and I just drew some dots here because this is 26, and I have 27, 28, 29. I don't know what those residues are, but it doesn't matter. I have arginine. So again, if you look here, you can see that we're going on this. So this is 26, 27, 28, 29, 30. So I'm kind of tracing this helix. These guys are on the same alpha helix. You get a nice ion pair between the carboxylate group and this guanidino group, which is positive. This is a little bit farther away, there might be an ion pair here. So here's a nitrogen. So this one you could go either way. I drew this beta chain protonated, in which case I have another ion pair here. But if you drew it unprotonated, because we don't really know the pKr of this, a free histidine would have a pKr of 6, but in the protein we don't actually know. So if you drew this unprotonated, this would just be a nitrogen. That could be a hydrogen bond. Oh, no, sorry, it wouldn't be over here. It'd have to be this guy. Yeah, you need a hydrogen bond donor, and you'd have to draw an interaction between here and the carboxylate group, which is a hydrogen bond acceptor. So I didn't draw it that way, but if you did, that was totally acceptable. Okay, so this is interesting. There is glutathione in red blood cells, and I actually didn't know about this until I read this paper, but apparently it can escape through a transport protein, a CFTR protein in red blood cells. So what these investigators did is they did an experiment to measure this. So if you look over here where it says basal, so these are the different treatments. On the y-axis, this is a measurement of glutathione efflux. So this is just a velocity. So it's the relative efflux of fluorescent conjugates per milligram of protein. Notice this is relative. So they would have measured that for just regular resting control cells. And since this is relative, whatever number that was, they just set it equal to 1. And then all of these are comparisons to that. So Here we have cells that are treated with hydrogen peroxide, so that's going to mimic the stress condition. And then these are GlyH101, MK571. I'm not sure where those names came from, but those are the names of the small compounds which are known to bind to the CFTR membrane transport protein. So we need to make something of these data. So if I take sort of a close look at these values, again, this is the control. If I add hydrogen peroxide, I get nearly a 6-fold efflux of glutathione. So it goes up almost 6-fold. If you add this compound right here, that efflux goes way down. It doesn't get back down to the control, but it does go way down. And this compound does the same thing. It's not quite as good. So the GlyH101 actually gets you down to basal a lot better than this compound, but both of them give you a decrease. So what you have to do when you're analyzing the data is note that, okay, here's my control, and again, if I mimic the stress conditions, I get about a 6-fold, almost a 6-fold increase in efflux. If I add this compound, I can decrease that by 4-fold. So if I go from here to here, that's about a 4-fold decrease. And if I go from here to here, that's about a 2 or 3-fold decrease. So the increase that I see with oxidative stress, I can get that to come way down if I add the GlyH101 and the MK571. Neither one of those brings it down to the level of control. And the GlyH101 appears to be better than the two because it brings down 4-fold rather than 2 to 3-fold. So what's happening here? Well, the only information that I have, other than the data that are presented, is that these agents bind to the transporter. So if they bind to the transporter, they must do something. We don't know what it is, but they must block the transporter somehow. We are given the information that they can bind. We are shown that when they're there, that efflux doesn't happen. So these show promise as drugs. So if you can get these inhibitors to bind to the transporter so that the efflux doesn't occur, then the effect of that is the glutathione doesn't leave, it stays in the cell. And that means it could react with the reactive oxygen species. And all of those things that the reactive oxygen species produce by the abnormal hemoglobin, all of those symptoms, you could counteract those effects. So these do have some promising use as therapeutic drugs. So if we compare lesioning with Rashmi, we don't have a lot of data for lesioning. We have more data for Rashmi. But the authors of this paper were able to make the case that she is better to adapt to severe anemia than other patients. She's in actually a much better shape than Li Jing. So is that true? And why is it true? So yes, it does appear to be true. If I go back to question 8 and I look at my Y values, we saw that Rashmi had her N value is low. So when her abnormal hemoglobin binds, the cooperativity is not quite as good. Her P50 value is also a little higher, which means that her oxygen binding affinity for her hemoglobin is not as high. But if you look at delivery, if you see how much oxygen is bound to these hemoglobins in the lungs and then how much is bound to the tissues, the difference, because we did this in recitation, if you remember, is, and we did this in class too with our case study, with our Memory case study, the difference is what's delivered. So if we look at these values, either Rashmi is a little bit better, or they're actually kind of about the same. But the point is, she doesn't have a problem with oxygen delivery. Her hemoglobin is able, or whatever combination of hemoglobin she has in her blood, she's able to bind oxygen in the lungs. So the affinity is great enough to do that. And the affinity decreases to the extent when the hemoglobin fully loaded with oxygen gets to the tissues, it can release it. So she seems to be in pretty good shape. So yes, we support this statement. So her ability to deliver oxygen in the tissues is either comparable or maybe even a little bit better. So we support the statement. So why do we think that is? Oh, my arrows don't look very good here. Sorry about that. Rashmi's hemoglobin definitely is a whole lot lower. She is anemic by a lot. If you look at the amount of hemoglobin she has, her P50 is higher. but she has a lot more BPG and she has a ton of fetal hemoglobin. So she's clearly anemic, but she has higher BPG and higher hemoglobin F. So we already noticed the difference in the P50. But the increase in BPG is interesting. The increase in hemoglobin F is also interesting. So we should use what we know about how BPG and hemoglobin F behave so we can figure out what's going on here. So let's look at hemoglobin F first. Fetal hemoglobin normally stops being made when the baby, after 6 months after a baby's born. But she's 5 and she's still making it. So hemoglobin F can form between 2 alpha and 2 gamma chains. This forms very nicely. So if if the gene expression that gamma chain is still being made, she has extra alpha chains that she can use that to make hemoglobin F. And that has a side benefit because remember the alpha chains aggregate and cause oxidative stress. So if you're kind of using them up to make hemoglobin F, then you have a little side benefit of decreasing that. Hemoglobin F normally has a higher oxygen binding affinity than hemoglobin A because it doesn't bind to BPG levels as well. But BPG is higher, so we'll get to that in a second. So again, you should have noted what we talked about in class, how BPG binds to the central cavity, and if we have a serine instead of histidine at 143, so there's a 143 histidine right there, there's another one right here. If both of these guys are replaced with serine, you have fewer ion pairs. So BPG doesn't bind as well. So that means that the fetal hemoglobin has a higher oxygen binding affinity. So this could explain why she has no problem binding oxygen in the lungs because she has some fetal hemoglobin that can do that. That's not what's doing in the fetus, but she's not a fetus. She's a 5-year-old girl. So, but the hemoglobin F is doing what it does in the fetus, which is not binding to BPG as well and increasing the oxygen binding affinity. So, well, what about BPG? Because her BPG levels are higher too. Well, BPG stabilizes the T form, so you have to explain how this works, and I'll do that in a second. So if the hemoglobin F increases binding affinity of hemoglobin to the lungs, and the increase in BPG increases the delivery of oxygen to the tissues, that could explain why she has the P50 that she does. And that could explain why her oxygen binding delivery doesn't appear to be compromised. So just a quick note on how BPG works. Remember, BPG binds in the central cavity. It's got 5 negative charges. The central cavity of the T form is lined with positive charges. You form those nice ion pairs. So the ion pairs stabilize the T form. So when you have BPG, the oxygen binding affinity is decreased. You can do this in the lab. You can show if you add PPG, the P50 goes up and the oxygen binding affinity goes down and you stabilize the T form. And that helps you deliver oxygen to the tissues. So one last thing to address in this question is that we do have that beta 26 that's in close contact with the beta ARG and the HISP. You put a lysine there, and that is going to break that interaction. In fact, it's going to repel it. What happens after that, we don't know. But structure function, that changes the primary sequence. If we change the IMFs, the secondary, tertiary, and quaternary structure would impact oxygen binding, delivery, and cooperativity. We saw the cooperativity was decreased. But she somehow seems to be rescued from that by having more hemoglobin F and BPG. So I hope that this has been helpful. And if you have any questions, come and see me. And thank you for your attention. And I will see you in class.